Hello everybody, my name is Alberto Scoma and I work at the University of Ghent in Belgium. Oh, here we go. And I'm going to talk about hydrocarbons degradation and under high hydrostatic pressure. So mainly what happens here. So one first little remark. What's an oil spill accident? So the most recent one that is kind of known, I guess, for everybody was the Deepwater Horizon spill in April 2010. We had a massive release of hydrocarbons all at once, equivalent to a little bit less than 1 million cubic meters. And also there was some methane, like more than 200,000 megatons. So if you try to have an idea how big is that, so that would make more than 300 Olympic-sized swimming pools filled up with oil, and the equivalent of 35 billion zeppelins filled up with methane in the sky. Can you imagine a sky like that? So a huge amount of hydrocarbons all together. So by now we know that the oil floats and it goes up in the air, of course. So the question is, why bother about deep sea? Well, oil drops. So recently they found that just here close to the well that exploded during the Deepwater Horizon spill, they found this purple thing is oil, and this is a crab moving. So actually some of the oil dropped down. How much? There are estimates, more or less 10% of all that oil that I said before is on the seafloor, and it covers an area between 8,000 square kilometers, according to who said that, of course. And this is at the level of 1.5 kilometers deep. So this is 150 bars pressure. How the oil drops down? Because of two effects mainly. One is seize knot and the other one is dispersion. So dispersion were largely used for the first time during this oil spill. These are components that try to clog all the oil together and eventually this goes down. And seize knot is a kind of mucus that is produced by algae once they are in contact with a massive amount of hydrocarbons. So some drops are formed and they slowly go down. So by now we know that oil may contaminate the surface, the sky, but also the deep sea. Now what happens to this kind of oil when it goes down? Well, actually nothing happens. So if you check the literature, you have plenty of studies on that they are really wonderful. I just pick up one here from 2002. They checked the alkanes profile, and in a marsh, this is not deep sea, but it's seafloor anyway, and they found that after 30 years of the spill, here was the West Falmouth oil spill, the alkanes profile did not change, and here we are talking about C13 to C25, so pretty easy to degrade, and nothing changed after 30 years. So the story is that once the oil goes down, it will stay there forever. So if you check the literature here about the hydrocarbons degradation and high pressure, nothing has been done in the lab. So you have 4 publications in the whole history of science. So cultivation of piezophilics with oil is pretty much unknown. And 2 are from the 70s and something more recent. So how pressure behaves. Every 10 meters of seawater, you have 1 bar more. So more or less at 1 kilometer, you are 100 bars. If you check these 4 publications, they found not much in terms of microbes. Here they tested Aeromonas, Pseudomonas, Vibrio, Marinobacter, Rhodococcus, and Sphingobium. They always use kind of C16 and pressures were between 120 and 500 bars. So here we come to the scene and we said, okay, let's take an uncontaminated sediment and see what happens if we put some oil in it and track the microbial shift. So what kind of bugs develop and what kind of microbial community do we have? So how this shift in the microbial community behaves, and is there a significant difference with respect to 1 bar if you increase the pressure? Are there special needs for piezophiles or for bacteria that are piezotolerant as well with respect to 1 bar, and are there some different dominant bacteria at high pressure? So what we did was to go with a military research vessel called the Belgica. Well, we are from Belgium, so no surprise. And we went off of Lisbon in this area called the Western Iberian margin. This is where Spain goes down a little bit. And we sampled in several regions, but especially here at the deep sea station, this is more or less 1 kilometer, so 100 bars. And we collected with this multi-core device sediment from down deep. So this one would be, for instance, a core sediment. This is the seafloor, and this is the subsea floor. And this is the water down there. Then back in the lab, we just enriched this sediment in different conditions. So the native pressure is 100 bars, but we wanted to make a little bit of a zoom with different pressures to see if different populations would develop. So we tested 1, 100, and 200 bars, equivalent to surface water, 1 kilometer and 2 kilometers. And we provided as the unique carbon source C20. Icosane is already solid at ambient pressure, so it's really hard. It's not just liquid like 16, so it would be even more hard to degrade, but it has a higher chance to drop down. And the enrichment was just incubations of 10 days that we repeated 9 times, so 90 days. Every 10 days, we stopped the reactor, we sampled 10%, we add a fresh medium, and we kept on growing. So just the cells, the microbes that were really growing on C20 alone would eventually develop. And we did that exactly the same also with C30. That is even, I would say, more solid, if that's a thing. So we ended up with, starting with 2 inocula at the beginning, having 6 different biomass streams at the end, with this just 2 carbon sources, unique carbon sources, either C30 or C20, and these 3 different pressures. So a little bit of results now. I always divide graphs in C20 or C30. So we added 2 controls, the black and white. These are reactors where we had either no inoculum or no carbon source. And if you don't have both, you can see that pH value stays pretty stable. So in this case, pH value is an indirect measure of carbon degradation because C20 or C30 were the only carbon source. And what you can see is normally that reactors at 1 bar, the red one or the green one, eventually go lower, meaning that the degradation is higher. And reactors at 100 or 200 bars more or less behave the same. In general, C20 was more degradable than C30. This may be explained if you check the cell number. Here we run a flow cytometry to have exact values. And with C30, we have very scattered during the enrichment, very scattered values, while with C20 that was a little bit more linear. And as I said before, at 1 bar you have more cells that actually gives us more degradation, while 100 and 200 bars somehow are at the same level. Despite this scattered behavior here with C30, you can say that the number here are the same, you have the same scale. So irrespective of the carbon source, you have more or less the same range of cells. The fact that C30 gave less reliable results may be explained by the fact that cells growing with C30 are not very hydrophobic. So this test is very easy. Whilst you grow on oil, cells kind of change the membrane structure, something we saw before in the presentation with unsaturated fatty acids on the membrane. And normally, if they are more hydrophobic, it means that they detach from the watery phase and go towards the oil. So they have access to the carbon. So the more they are hydrophobic, the more easily they can degrade the oil. And with C20, indeed, you see that you have positive values during the enrichment at the end, while with C30, we had no connection. So normally, C30 is degradable, but it's very hard, and cells do not really develop an hydrophobic membrane. Nevertheless, we found some interesting correlation with nutrients. So we checked sulfates and phosphates. With sulfates, we had actually no relation because the reactors were all aerobic. In the case of phosphates, we have seen that, this is per cell, the consumption of phosphate increases and is statistically relevant at 100 bars and 200 bars with C20. This kind of happened also with C30, even though because of the scattered cell number, this values goes up and down. Please note that this is not standard deviation. This is a statistical analysis with a 95% confidence interval. So it's wider, but you can see a trend going up, and with C20, where you had more reliable values, you can see that phosphates are needed at high pressure. And what is interesting is that if you check the composition of the sediment of the inocula, you have no phosphates there. So if you imagine in that place where I said out of Lisbon there is a contamination with oil and there is no phosphates, this culture will not grow and they will not be able to degrade the oil as well, because we know that deep sea is normally oligotrophic. You don't have many nutrients. Well, in this case, phosphates are really needed. And coming to the microbial structure, we run Illumina sequencing to see exactly what happened. So this is kind of given information. We all know that after a contamination, there is a microbial shift. What is interesting to me is that we observed within the days of enrichment, and with the first incubation, the first 10 days, we lost 1 order of magnitude of biodiversity. And within 30 days, probably even before, because I don't have here a 20 days point, we were already stable with a loss of biodiversity of something like 95%. So meaning that just 3% of the initial biodiversity is kept, the rest is lost. And during the enrichment, this stays pretty stable. So if you don't remove the oil from there, you will lose this biodiversity forever. We did some DGG as well to see how the community shifted in a kind of rough way. And already halfway after 50 days, here you can see that clearly, that the community was already stable. Because we used 3 different pressures, 100, 200 bars, this kind of zoom allowed us to have a little bit of understanding of the different groups of bacteria. Normally you talk about piezo-sensitive, piezo-tolerant, and piezophiles. Piezo-sensitive are those that cannot cope with pressure. It's the red square, and you just see them at here at 1 bar, and you don't have these bands if you increase the pressure. Piezo-tolerant are those that actually can grow also with higher pressure, so you normally find them everywhere. Actually, the graph is not very good here. And piezophiles are those that can only grow with pressure, so they need pressure. You don't find them at 1 bar. So by applying these different pressures, we could zoom a little bit. This means that the initial inocula had a little bit of everything, but if you apply different conditions, the structure of the microbial structure will differ as well. At the end of the enrichment, as I said, we had 2 inocula and 6 biomass streams at the very end. They were enriched. And if you check the composition, more or less you have the same genera, but the structure of the 6 biomasses we obtained was different. And if you zoom a little bit more, we saw something interesting. That is, at 1 bar you have just red and green, irrespective of the carbon source, and these are alpha and gamma proteobacteria. They dominate at 1 bar. But if you increase the pressure, then also the blue and the yellow pops up. There are the delta and epsilon proteobacteria. So you have different communities. If you increase the pressure, they do not enrich, do not develop at 1 bar. If you check a little bit more closer, we found that piezo-sensitive strains, just growing only at 1 bar, are something like Alcanivorax that you can see here, that disappears if you increase the pressure. On the other way around, we've seen like Sulforospirillum or Photobacterium just growing at high pressure. And piezo-tolerant would be like Pseudotoromonas that you can find in any of the enrichment. So these are the names that pop up in the end. So to summarize and conclude, how does the microbial community shift over time? We have seen that within 30 days, the biodiversity loss is incredible and is already stable, meaning that you end up with 3% of the biodiversity you had at the beginning. Does the microbial community structure differ at 1 bar? Yes, because if you increase the pressure, you will always have delta and epsilon proteobacteria that do not grow at 1 bar. What are the special needs? We have seen that if you increase the pressure, you need more phosphates. And that's an issue at deep sea because the deep sea sediment is not rich in phosphates. And then when it comes to the dominant bacteria, if you increase the pressure, we found Desulfovibrio, Desulforomonas, Photobacterium, and unclassified delta proteobacterium, and Sulforospirillum. Why is this interesting? Because if you check the publication until now, you don't have them. So these are completely different. That's also kind of story. If you keep on enriching or studying the same carbon source, you end up with the same strains. So you have to differentiate a bit. Here we use C20 and C30, and that's what we managed to have. What's next? We are isolating, because now I really want to have these bugs in my hand and try to do some synthetic communities and see how function and structure behaves. And we run some metaproteomics of the enrichments, and this is done in collaboration with Professor Bendorf at Magdeburg University. And this is really all. I thank you for your attention.
Thank you, Alberto. Right in time. Questions? I can take two. Yes, I have two. Oh, sorry.
So I have a question according to the occurrence of phosphate down in the deep sea. Did you analyze the minerals in the deep sea floor, for example? And why you add oxygen to your cultures, and why you don't oxygen, for example, some sulfur?
So about the minerals, we run some very basic analysis, nothing too serious, I would say. Here we just check chlorides, nitrates, nitrites, phosphates, and sulfates. And these are the values we obtain. Not determined, meaning that the lower level is 0.2 milligrams per liter. So you have less than that. And that's the basic analysis we've done. We didn't go too much deep in that. And coming to the oxygen, degradation of C20 and C30 is really hard. So I wanted to have oxygen, and indeed the sediment that I picked up is the first 2 centimeters that are still kind of aerobic. Because if you go anaerobic, then it would take even longer. I really wanted to have a fast enrichment because I had no idea whatsoever if the bacteria could grow on C20 and C30. So I kept everything aerobic. At 1 bar, it was easy because I just increased the headspace. With 100 and 200 bars, we first provided 30 bars air, and then we brought to pressure with liquid up to 100 or 200. But if you go, of course, during the core, along the core of sediment, the first part is aerobic, the rest is not. So degradation of oil below the subsea floor, in the subsea floor, it will be even longer than that. So, and here I did not report on any degradation kinetic, but 90%, 95% of what we gave was still there. So I checked activity, pH goes down, but there's a lot of carbon that you put there. So if you want to study something with sulfate, of course, many of the bacteria that I showed are all sulfur related. So we're talking about the sulfovibrio, the sulfomonas, and so on. Okay, sorry.
I have a quick question. Nice
Yeah.
talk. Can you may explain your shift in the biodiversity based on the cell wall? Not meaning that the cell wall is more thick or less thick, but maybe the transport system to get this 20C or 13C inside the cell is made different between the 2 types of diversity. Is this maybe an explanation?
No, I would not say so. So the increase of pressure brings normally unsaturation on the membrane, because the unsaturation of the PLFA brings a lot of buffering activity, so the membrane can contract a little bit. But the degradation and the activation of the hydrocarbons occurs on the membrane, and after you have an import. So I don't see the dimension of the hydrocarbon has an effect. Pressure is much more important. And for that, I think that because they all belong to 100 bars, they all have already unsaturated membranes. So that will be the same for all the bugs.
Can continue later.
Yes.
Thank you very much.
Thanks.